Practical is able to deal with the different stages of mitosis, and you have to be able to do these slides. We learned about mitosis at 10th standard. With this time, you're going to sit and do the stages of mitosis, and you're going to do it with the help of the following experiment. Here, what I've done is, long ago, I've taken an onion, pierced it with a needle, and kept it in a beaker containing water so that I can develop roots. So you can see the roots here. Now I did the whole root here, but I need the tips of the root, because in the tips of the root there will be meristematic tissue. Wow. And in plants, the only place where cell division occurs is in meristematic tissues. So now I have to gather some of the meristematic tissues from the root tips. I have to collect that and proceed as follows. Now in this step I have to find the different stages of mitosis. The procedure is simple. You need certain things. First, you need an onion bulb, and you need a pair of needles. These are the pair of needles. And then you need slides. Glass slides, you need filter paper. You need a test tube holder. You need brushes, and you also need a spirit lamp. And then you need a dye called acetocarmine. And then I need some HCl also, and a spirit lamp. Okay. Now what I'm going to do is, I'm going to take a few of the root tips. I cut them off and place it in a watch glass. Now what I'm going to do is, I'm going to keep the root tips here, and I'm going to cut off the tips. Notice, I'm taking only the tips. I'm not taking anything else. I don't need the other part because you won't have cell division in those parts. Only the meristematic tissue, and the meristematic tissue is present only in the root tips. I don't need all. I don't need anything in the onion except the root tip. I have a few root tips now. So I am going to discard this onion. And notice here, you can clearly see that I have a few root tips. You see this? Now I am going to treat these root tips as follows. I am going to add 9 drops of acetocarmine solution. Stop. Continue to see this. We are going to light the spirit lamp now. We need to hit this. Okay, what we are going to do is, we have a spirit lamp. This is called. Okay, so here what we are going to do is, you see I have my root tips here. Now to these root tips, I am going to add 9 drops of this dye. Please check this. And this is called acetocarmine. This dye, you take 9 drops, exactly 9 drops. Do not mix up things, be very careful. This is a very important part of the procedure. Count with me. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 drops of acetocarmine and 1 drop of HCl. Acetocarmine is the dye that actually helps in coloring the cells, but HCl is meant to break the cell walls. So that is the reason why we use this mixture. Always ask yourself, why did I use this combination? Now once you finish that, keep it aside and use your test tube holder. Hold the watch glass like this. Make sure your root tips are inside the mixture. Mix it properly. Now this is what I have. And now I'm going to hit this. How much to hit? Till it starts fuming. Do not overheat till it starts fuming. As it starts fuming, take it out and don't inhale the fumes. You'll get a terrible headache. Okay, just be careful. This is what we are going to do. Okay, now I see some fumes coming out. Now keep it aside. Now once that happens, keep it aside and collect your leave the mixture aside for 1 or 2 minutes. Now the spirit lamp work is over. So put it up. Okay, so not like that. I mean, you have to put a cover. I don't have a cover, so I did that. Don't try that at home. Okay, so okay, just take take a glass light. This is called a glass light. Okay, and you put 1 or 2 of your root tips on top of it here like this. The root tip is now placed on this. Okay, and now after this, keep a filter paper under your slide. Where is that? Okay, come here, filter paper. Alright. Now see, I have this here. Now take half the drop of acetocarmine and put it on that. Okay. Now what we do is, where's my blade? Here's my blade. Now what we're going to do is, we're going to chop, and this is called macerate. Okay, that's what you're going to do. You're going to macerate. Okay, macerate means to cut into tiny pieces. Best use the edge of the blade and go smashing. Think of Hulk Hogan and hammer everything. Okay. Just smash everything like Hulk. Okay, keep smashing. The more you smash, the better. And this should be like a paste. Now you either get it or you don't get it. As simple as that. So just make sure that you get it, because you get one attempt at doing this. You won't have much time left in the exam if you're going to do this for the exam. And now when you mash it, the mixture should look somewhat like that. And just see that, so you can't see that. Now see, you keep a background and see. You can always use a background, okay? And I keep doing it. I'm not happy with this. I need some more. So I need to put half a drop. Don't put too much dye. It will flow away. So I put half a drop. Half a drop. Be economical here. Don't just pour a lot just because there is which are available. Now keep doing that. Keep doing that. This needs patience. As you keep doing this, the more you squash it, the better it will be, and the cells will spread out. Best do these experiments in the early morning, because mitosis in plants occurs early morning. So basically, it's a good idea to do this experiment in the morning. Okay, now I've got a mashed up tissue here. The more I mash it, the better. This takes patience. Do not hurry up. You won't get any prizes for finishing first. You finish last, no problem. But make sure that you get some stages at least, because if you don't get stages, all the work that you did is a waste of time. We want productive work, not hard work. You can keep your hard work to yourself. Give us productive work. Okay, now I've got master tissue. I've done a good job, even though I only am saying it. This is what you did. Once you get that, now you come to this box. This tiny little box is known as a cover slip. Here, what I'm going to do is I am going to take one single cover slip. Be very careful. They look tiny little pieces of glass, but they are seriously expensive. They can cost up to 10,000 rupees for a unit. So just be very careful. Now what you do is slowly, where's my needle? See, this is a needle. Okay, place your cover slip on the needle and come closer, please. Okay. So when you place the cover slip on the needle and pull your needle away slowly so that no air bubble gets into your slide. That is the technique. Now take this slide, keep it aside. Fold your filter paper like that. Okay, once and twice when you fold it like that. Now take your slide and place your slide in between like that and press gently. Do not press with all your force. You're all strong people. You'll break everything. Don't just press, because the excess of dye will flow out. Now that is why you do this. To take away the excess dye, and don't think more colorful slides give you more marks. More colorful, less marks. You can also get 0. Be careful. This is what you do. Now once you do this, your slide is ready for viewing under the microscope. Now this is what we are going to do. Now that is a slide which is ready made, and it has certain stages. I'm 100% sure, because I took the proper tissue. Sometimes when you take a tissue, you may not get stages. Now I want to see the stages of mitosis here, and I'm sure you all learned this. See, I have a sample record here. My dear friend has written this record, and it doesn't matter. I purposely am holding it upside down. I showed her plant cells. See the diagram she's written. She's written all animal cells here. Don't do this mistake. That's why I'm holding it upside down. This kind of work is sloppy and is done for the heck of it. I want you to draw a plant cell, and you all know a plant cell is rectangular, and you all saw that I took it from the root of a plant. How can the cell be circular? And you try telling them, they argue with you. Excuse me, let me focus it for you under the microscope. When you focus it under the microscope, this is what you do. First, the microscope is like that. There is a lesson on microscope. I'm combining it here. Now guys, this is called the eyepiece. This is called the objective. And this part is called the arm of the microscope. This is the foot of the microscope. Anytime you want to lift the microscope, hold it by the arm and lift it. Now, this is, okay, there are two types of lenses. One is a small lens. The other is a long lens. The small one is low power and the long one is high power. Now, whenever you want to bring this into light, make sure that you rotate this disc and you hear a click. When you hear that click, that means the eyepiece, the objective, and the mirror over here are all in the same straight line. And you need this in order to focus something. Now this here, the big one, is called the coarse adjustment. Now when you move this up and down, the microscope moves a lot, the eyepiece moves a lot, but this is also called fine adjustment, and this fine adjustment allows very tiny movement. Okay, this is for sharp focus, this is for coarse focus, and this clip here, this clip is meant to hold the slide, but unfortunately the clip has gone for a walk, so there is no clip here. Then you have a mirror here. The mirror is required for focusing light, and there are two types of mirror. If you notice, one is a plane mirror. Feel that with your finger? It's a plane mirror. On the opposite side, you have a concave mirror. Now, this concave mirror is used when you're using the microscope indoors, whereas when you're using the microscope outdoors, you use the plane mirror. And this is what happens. The first thing that you do when you get a microscope is move the microscope towards the source of light. Adjust it in such a way that the eyepiece, the objective, and the mirror are in the same line. How can you do that? You can do that by checking for the amount of light that is entering the microscope. If there is bright light, that means your parts are in line with each other and you can proceed with your work. If not, move the object here. See here, now it is not in line because I have moved the object. Now just move it slightly till you hear a click. Can you hear that? Now when the click happens, the microscope comes in line. All the three parts, the eyepiece, the objective, and the mirror are in straight line. So now you can focus your eyes. Now take the slide we made sometime back. Now use this slide, put it under the microscope and see it under the microscope for the different stages of mitosis. The stages will appear as follows. Thank you.